रेस्पिरेशन टॉपिकच्या अंडर आपण 8.3 पर्यंत आलेलो आहोत. 8.3 म्हणजे मेकॅनिझम ऑफ रेस्पिरेशन. आता पुढे आपल्याला जायचं आहे 8.4 दॅट इज रेग्युलेशन ऑफ ब्रिदिंग. तत्पूर्वी तुम्ही जो सगळा अभ्यास आहे तो पूर्ण केलेलाच असेल अशी अपेक्षा आहे मला. आता आपण रेग्युलेशन ऑफ ब्रिदिंगकडे जाऊया. नाउ वी हॅव स्टडीड mechanism of breathing by two ways, inspiration and expiration. Now you are familiar. During inspiration and expiration, various regions of thoracic cavity, that means sternum, diaphragm, intercostal muscles, etc., and the lungs, they involve in inspiration and expiration. Overall, that is considered as a breathing. Now, human adult, for your information, human adult can breathe about 12 times per minute, whereas newborn can breathe about 44 times per minute. Remember these figures because that will be asked in MCQ. Now this breathing is controlled by two ways. These two ways are nervous control and chemical control. Nervous control means in that control the brain actually involves. You can see in this figure also. Now along with the explanation we'll move to the figure also. So nervous control involves brain. Chemical control involves the concentration of CO2 or partial pressure of CO2 and concentration of H ions. How? We'll summarize one by one. Now first of all, nervous control. Now central nervous system contains pons moruli and medulla oblongata. These are the two regions of hindbrain. Complete central nervous system is divided into three regions: forebrain, midbrain and hindbrain. Out of these three, the hindbrain shows two regions, pons morula and medulla oblongata. In the figure you can see this is brain and this is pons morula and this is medulla oblongata. These pons and medulla together actually controls the steady rate and depth of a respiration. What is meant by steady rate? The 12 times per minute or 44 times per minute in a newborn. And the depth of respiration, that means complete lungs should be filled with air containing oxygen, and at the same time or the next cycle, expiration, during which the lungs will be expired the air containing CO2. We have already studied the lung volumes also. So steady rate and depth of respiration is controlled by both these pons and medulla oblongata. Hence, they are collectively known as respiratory centers or RC. Respiratory centers or RC. These respiratory centers contains three regions or group of neurons. The first is dorsal group of neurons or ganglion, or it is also known as DRG. D for dorsal, R for group and G for ganglion. This DRG is present in medulla and it is the inspiratory center. Second group, ventral group of neurons or ganglion. In short, it is known as VRG. V for ventral, R for group, G for ganglion, VRG. This is present lateroventrally in medulla. The DRG is present dorsally, that's why the name is given dorsal, whereas ventral group or VRG is present ventrolaterally in medulla. And most of this respiratory center is expiratory center. DRG is inspiratory, VRG is expiratory. But in our textbook, additionally, here inspiratory center is also mentioned. The third group is pneumotaxic center. And this word pneumotaxic, P is silent. Remember, do not pronounce the P. Start pronunciation from N. That's why pneumotaxic center. So third group of ganglions known as pneumotaxic center, this is present in actually pons. Pons borrowla, you can see here. Both collectively these are known as brain stem. The pons is present above and medulla is present below, forming the stem of brain. That's why they are collectively known as a parts of brain stem. This pneumotaxic center present in pons, actually it functions as limits inspiration. Limits inspiration means how much amount of air should be entered in lungs. What is the capacity of lung? You know very well, maximum capacity of lung is 5800 ml. Up till that capacity, lungs can becomes full of air. So limits inspiration, slow wave sleep and rapid movement sleep. That means at the time of sleep, the sleep is slowed down, the sleep wave is slowed down by rapid eye movement sleep, and that function is controlled by this pneumotaxic center. So now you are clarified. The respiratory centers that are present in pons borrowla and medulla oblongata. Both of them, they control steady rate and depth of respiration. They include three groups. First is DRG, second is VRG, third is pneumotaxic center. DRG is inspiratory center present in medulla. VRG is expiratory center, but in addition inspiratory also to some extent, present lateroventrally in medulla. And pneumotaxic center present in pons. So all these groups they are collectively known as respiratory centers. One thing is there are another centers also present exactly opposite to the pneumotaxic center known as apneumotaxic centers. These apneumotaxic centers, they are also present in medulla and they are opposite to pneumotaxic centers present in pons. So medulla contains apneumotaxic centers and pons contains pneumotaxic centers, and they are opposite to each other because it controls non-rapid eye movement and sleep and wakefulness. That means this apneumotaxic center brings about the waking from sleep or non-rapid eye movement. During sleep, there is always rapid eye movement occurs, and during wakefulness, always there is non-rapid eye movement occurs, or in another word, the rapid eye movements slows down, and we get we wakes up from sleep. So remember this point also. Aneustic centers in medulla are opposite to pneumotaxic centers of pons because it controls non-rapid eye movement, sleep and wakefulness. Now actually how this process occurs? This process is also summarized as Hearing-Brewer reflex. Hearing-Brewer reflex, the name of scientists. What happens, see, during respiration, lung expand. Now you are well known. During inspiration, lung expands. And due to expansion of lungs, stretch receptors stimulate and present on the lung alveoli, and nerve impulse are sent along the 10th cranial nerve vagus to the VRG, that is expiratory centers. So during inspiration, the lungs expand and stretch receptors now get stimulated because of expansion, stretching, and that nerve impulse initiate and sent along this vagus nerve to the VRG. So you can see here also. Then the inhibitory impulses to DRG. Continuously, the vagus nerve first send nerve impulses to VRG, that is expiratory centers, then to the inhibitory impulses to DRG, that is inspiratory center, and inspiratory muscles relax. That means which muscle actually? Intercostal muscles. Intercostal muscles. Now in this diagram of regulation of breathing, which is given in our textbook, the brain, the thoracic cage and the heart is drawn. And just I have told you where pons florali is present, where medulla oblongata is present. Both of them are collectively known as respiratory centers, right? And here, this is thoracic cage containing sternum at the center. Then these are intercostal muscles, these are ribs, this is diaphragm. The nerve innervating to intercostal muscle, send the nerve impulse. This is another nerve, phrenic nerve, sent to diaphragm. And you know very well, during expiration and inspiration, diaphragms involve, and the nerve impulses goes respectively by this process. So, the inspiratory muscles relax and expiration occur. After expiration, the lungs now deflates. Deflates means air is thrown out and lungs becomes very small in size, thoracic volume decrease, thoracic cage comes into its original position and so on. You know very well. So due to deflation of lungs, stretch receptors now no longer stimulated. In first step, the lungs are expanded and due to which stretch receptors are stimulated, and that's why nerve impulse is started through this vagus nerve and goes to VRG, then to the DRG and inspiratory muscles, and relax and expiration occur. But after expiration, this signal, nerve impulse signal, not stimulates because lungs becomes flattened up, deflates, air is thrown out, and again the new respiration begins. That means again new cycle begins. These events are collectively known as Hearing-Brewer reflex. So in short, Hearing-Brewer reflex controls the depth and rhythm of respiration, and due to this reflex, the bursting, that means overstretching of lungs is prevented or avoided, and also due to overstretching, the lungs may burst and that bursting is also avoided due to this reflex. Yeah? So that is the process how this regulation of breathing occur by this nervous control. Now, some more points are also there. The pons and medulla, that means respiratory centers, they have also connection with cerebral cortex. That means here, the cerebrum, you know very well, the brain has 85% region occupied by cerebrum, and that cerebrum has outer surface known as cerebral cortex. So these pons and medulla, they are in connection with the cerebral cortex and they bring about voluntary breathing. They bring about voluntary breathing, because here this breathing is actually involuntary process. That point is remaining. The breathing is actually involuntary process, but pons and medulla, which are performing involuntary function, they are also taking participation in voluntary breathing also by connecting themselves with cerebral cortex. This voluntary breathing is protective in nature. That means how the voluntary breathing occurs as per our will. When we stop breathing and start breathing, for example, to avoid entry of water inside the lungs, or there are some irritating gases entering inside the lungs, we voluntarily stop the breathing, and that voluntary breathing is in connection with this cerebral cortex or in coordination with cerebral cortex. So also one more point is there. This centers, cerebral cortex centers, along with connection with pons and medulla, has ability to stop breathing, because when inside the blood concentration of CO2 increases. If concentration of CO2 increases, ultimately the breathing stops. That means we have to expel air outside, because our blood contains lots of carbon dioxide. So here we complete the first point, nervous control, under which we have studied these respiratory center, brain, respiratory center, pons corolla and medulla oblongata. This is thoracic cage. Thoracic cage contains intercostal muscles, ribs, diaphragm. Here there are DRG and VRG, and in pons there are pneumotaxic centers are present. In medulla there are DRG and VRG are present, and they are supplied with through these nerves, that is intercostal nerve and phrenic nerve. Also phrenic nerve goes to diaphragm. So here these nerve impulses start contraction of intercostal muscles during inspiration or during breathing, collectively you can say. Now we'll go to the second part, and the second part is chemical control. Chemical control. Actually, in our textbook, both these are mentioned, but the explanation of this chemical control is not given. Remember. So we have to explain this chemical control in addition. Remember, it is not given in the textbook. So listen carefully. What is the chemical control of breathing? This chemical control of breathing is entirely depends on concentration of CO2 and H ions. Completely depends upon this concentration of CO2 and H ions. Actually not depends upon oxygen. Or in another word, oxygen not have any significant effect there for regulation of breathing. What happens, see, this concentration of carbon dioxide and H ions and their effects when inside the blood, the concentration of CO2 and H ion increases, either CO2 or H ions increases, it affects on the receptors present on the carotid artery and aortic arches of heart. Now what is meant by carotid artery and what is meant by aortic arches? See here, you can see this is heart, auricles, ventricles, this is systemic aorta, this is pulmonary aorta. These aortae, their walls are thick, and these aortae containing walls, thick walls or having thick walls, contains bodies. These bodies are called as aortic bodies. These are called as aortic bodies, and they contain receptors. The carotid artery and aortic arches, they contains bodies and they shows presence of these receptors. These receptors are known as chemoreceptors. Ultimately, the chemoreceptors are also seen in medulla. Medulla also contains chemoreceptors. When blood is flowing through circulation and reaches up to this carotid artery and aortic arches, the bodies containing these receptors get stimulated. The nerve impulse is created that there is a higher concentration of either PPO2, you can see high PPO2 and H ion concentration, because they are highly sensitive. They are highly sensitive to either partial pressure of CO2 and partial pressure of or concentration of H ions. So when inside the blood flowing through circulation reaches up to carotid artery and aortic arches, these receptors get stimulated and they are highly sensitive. This sensitive nature of this carotid body and aortic arches creates signal, nerve impulse signal, and sent to the chemosensitive area of medulla. Because medulla also contains, you can see in this figure, these centers, aortic bodies and carotid arteries, they send signal to medulla, right? And ultimately, the respective changes they are bring about, that means expiration, bring about with the help of this chemical control of breathing. So in this figure, you can see this part of figure shows nervous control, purely nervous control. This part, that means heart, shows chemical control along with nervous control. Or in another word, nerve impulse is initiated and then goes to medulla and bring about the certain necessary changes. Right? So here we complete the 8.4 unit regulation of breathing. Right? I will summarize in brief. The regulation of breathing, for example, adult breathes 12 times per minute, newborn breathes 44 times per minute, and this breathing is purely involuntary process, controlled by two ways, nervous control and chemical control. Central nervous system contains pons horula and medulla oblongata. They controls the steady rate and depth of respiration and hence called respiratory centers. These are three: DRG, VRG and pons. DRG is inspiratory center, VRG is expiratory center, whereas pons is limiting inspiration, slow wave sleep, rapid eye movements, sleep, etc. And aneustic centers in medulla are opposite to pneumotaxic centers of pons, because these aneustic centers, they are responsible for wakefulness and controls non-rapid eye movement. And that process is summarized by hearing Brewer's reflex. During inspiration, lung expand, stretch, stimulate nerve impulse, send along this vagus nerve to ERG, then to DRG, then inspiratory muscles relax, expiration occur, lungs becomes empty, and stretch receptors are now no longer stimulated, hence new respiration begins. This is called as hearing Brewer reflex. This hearing Brewer reflex controls depth and rhythm and respiration, depth and rhythm of respiration, and prevents busting of lungs. So this is involuntary process. Voluntary breathing also occurs because it is protective in function. Pons and medulla have connection with cerebral cortex and brings about voluntary breathing. For example, avoid water entry, irritating gas entry in the lung, and also stop breathing because when concentration of CO2 is high in blood, that is called as voluntary breathing. So that is studied with the diagram. Then chemical control is completely depend upon concentration of CO2 and concentration of H ions in the blood. These are detected by receptors present on carotid artery and aortic artery, and they stimulates nerve impulse. That nerve impulse goes to the chemosensitive area of medulla and brings about the rhythmic respiration. So here we complete the 8.4 regulation of breathing. Clear?